Hi everyone, Kelton here. Today we are going to be discussing one of my favorite topics, the human heart, the structures, and its blood flow patterns. So pause the video, look at the outline, and we're going to jump right in. To start off, how many chambers are in the human heart? And great, so you probably already knew that a human heart has 4 chambers, but this demonstrates the trend that we looked at last video really well. As an organism increases in complexity, so does the complexity of its heart. And remember, a more complex heart has more chambers, so it only makes sense that something as complex as a human has a 4-chambered heart, while a frog has a 3-chambered heart. Our hearts are typically divided into left and right halves, and that's because if we draw a line right down the middle of the heart, we find that the chambers on the right side, so the right atrium and the right ventricle, are typically filled with oxygen-poor blood. On the left side of the heart, we see a left atrium and a left ventricle, and these are filled with oxygen-rich blood. Both the left and the right sides of the heart have very different functions, and we'll talk about them more as we go on. Whenever I study the circulatory system, I like to think of myself as a red blood cell that was dropped off in the body somewhere, and my job is to describe everything that I see along my way back to the heart. So let's say that we're a red blood cell that's just dropped off some oxygen to the gut and is now making its way back to the heart. We're going to be traveling through a series of veins until eventually we're going to enter a large vein called the inferior vena cava. Despite having different names, the inferior and superior vena cava have the same job, and that job is to return oxygen-poor blood to the heart. The superior vena cava has the job of returning oxygen-poor blood from above the heart, while the inferior vena cava here has the job of returning oxygen-poor blood from below the heart. From the inferior vena cava, we next travel to the right atrium. I like to think of the right atrium as the receiving room for all the oxygen-poor blood in the body. In other words, the blood that isn't coming from the pulmonary circuit. Once the right atrium contracts, we are then going to pass a valve. This valve can be seen here, and it's known as the tricuspid valve. It's called the tricuspid valve because it has 3 cusps, as we can see here. The tricuspid valve is also referred to as the right atrioventricular valve. The way that I always remember that the tricuspid valve is on the right side of the heart is by this mnemonic here. Tricuspid is on the right. Both atrioventricular valves have the job of closing when the ventricle begins to contract. This means that there's no backflow that can occur. Backflow is basically just if we had blood from the right ventricle make its way back into the right atrium, which wouldn't be ideal. So this begs the question then, how do these valves stay closed when they're supposed to be? Nice, so the shape of the valve itself is largely responsible for why this valve doesn't open during aventricular contraction. If we look at the valve here, notice that it almost looks like an inverted parachute. If the pressure from the ventricle is pushing it out, it's going to push this valve more closed. The next thing that helps the valve stay sealed during a contraction are muscles known as papillary muscles. So these papillary muscles are connected to the valve itself through tiny little tendinous attachments known as chordae tendineae. These papillary muscles contract at the same time as the ventricles in order to prevent the atrioventricular valves from prolapsing, or in other words, opening up in the wrong direction and allowing blood flow through. Once we pass the tricuspid valve, we are now in the right ventricle, which is basically just a sending room for oxygen-poor blood. This demonstrates that the right side of the heart is the oxygen-poor side of the heart. So notice also that the right ventricle's walls are much more muscular than that of the right atrium. This is because the right ventricle has to pump blood out of the heart, while the right atrium only has to pump blood down to the right ventricle. In order to leave the right ventricle, we have to pass through our pulmonary semilunar valve. Notice that both of the semilunar valves can be seen here, and they both have 3 cusps. The purpose of the pulmonary semilunar valve is to make sure that no blood can flow back from this vessel into our right ventricle. Once we've passed the pulmonary semilunar valve, we are now heading into the pulmonary arteries. The pulmonary arteries are going to carry oxygen-poor blood towards the lungs where the blood can then be oxygenated. This poses a question then that a lot of people typically miss. What is the difference between an artery and a vein? So you probably have heard at least one time in your heart that veins carry oxygen-poor blood while arteries carry oxygen-rich blood. Well, that's not really the case all the time, and we just demonstrated that because the pulmonary arteries are carrying oxygen-poor blood. Instead, we want to think about arteries and veins in terms of the direction of flow. Arteries are going to pump blood away from the heart. I always think away. Veins, on the other hand, are always going to be carrying blood back towards the heart. So now that we've picked up some oxygen at the lungs, we're going to be traveling back to the heart through a series of vessels known as pulmonary veins. From the pulmonary veins, we are going to enter into the left atrium of the heart. The left atrium of the heart is just going to act as a receiving room for oxygen-rich blood. Once the left atrium begins to contract, we're going to pass by another valve, the bicuspid valve. The bicuspid valve can be seen here, and notice that it has two cusps. This valve is also known as the mitral valve, and it's sometimes even referred to as a left atrioventricular valve. The bicuspid valve functions similarly to the tricuspid valve, and then it just prevents backflow of blood from the left ventricle to the left atrium. It's also important to note that just like the tricuspid valve, the bicuspid valve also has papillary muscles with chordae tendineae. After we pass through the bicuspid valve, we are now in the left ventricle. The left ventricle is just going to be the sending room for oxygen-rich blood, and this is going to be the most muscular chamber of the heart. The reason that the left ventricle is so much more muscular is because it's going to be sending its blood to the systemic circuit, and in the systemic circuit there's a lot of resistance, so it has to be a very powerful contraction to get the ball rolling. This is a really good time to note that even though some chambers are more muscular than other chambers, all of the chambers hold the same volume of blood. If they don't hold the same volume of blood, this leads to a lot of problems. Once the ventricle begins to contract, we're going to pass through yet another semilunar valve known as the aortic semilunar valve. The aortic semilunar valve has three cups just like the pulmonary semilunar valve, and it has the same job of preventing backflow of blood into the ventricle. After we've passed the aortic semilunar valve, we are now in the largest artery in the body, the aorta. The aorta also has the highest blood pressure of any vessel in the body. This is because all of the blood from the ventricle is pumped into a single vessel, and the aorta has a lot of resistance due to the systemic capillaries that it's pushing blood towards. So now that we've made it through this much, I want you to try to practice this on your own. Imagine you're a red blood cell in the lower half of your body, and you're trying to make your way to the aorta. Describe any of the features we've talked about today that you might see along the way. Okay, so if we're a red blood cell that's returning from below the heart, we're going to return to the heart through the inferior vena cava, sometimes labeled as IVC. From here, the blood is going to enter into our right atrium. Once the right atrium contracts, we're going to end up passing by our tricuspid valve, which I'll label as TV. We're also going to see that on the tricuspid valve we see chordae tendineae attached, and these chordae tendineae are anchored to papillary muscles. From here then we enter into our right ventricle, and within the right ventricle we are going to see our pulmonary semilunar valve. Once the ventricle contracts and we pass through the pulmonary semilunar valve, we're going to end up passing into our pulmonary arteries, and then we're going to end up at the lungs. After we've gotten our oxygen then, we are going to return to the heart via our pulmonary veins. The pulmonary veins then will lead us into our left atrium, and this is going to make up the entire pulmonary circuit. This is the circuit that describes how blood is going to be oxygenated. Once our left atrium begins to contract, we're going to end up passing our bicuspid valve, which we'll label as BV. Remember, this also has chordae tendineae and papillary muscle present. And after we've passed this valve then, we are officially in our left ventricle. Once our left ventricle begins contracting, we're going to pass by our aortic semilunar valve, and we're going to enter into the aorta. From the aorta, we're going to go nourish the body's tissues, and once we've lost our oxygen, we're going to travel through veins back to the inferior vena cava. The last half of this journey from the oxygen-rich blood in the left atrium to the body's tissues and then back to the right atrium as oxygen-poor blood is all part of what we call the systemic circuit. This is a really great way to practice what we learned today. And as we begin to add more information to the circulatory system, we can describe even more things as a red blood cell. So it's a really effective study tool to study this way. So now that we've finished with the lesson, let's go ahead and do a mini quiz. Which structure is in contact with oxygen-rich blood? So right off the bat, we know that we're going to be looking for something from the left side of the heart because it's oxygen-rich. So we can go ahead and get rid of any of our right-sided heart structures, like the right atrium is a pretty obvious one, and the vessel that it's connected to, which is the inferior vena cava. This leaves us with the choice of either pulmonary arteries or pulmonary veins. Remember, pulmonary arteries are going to take blood from the right ventricle to the lungs to be oxygenated, while the pulmonary veins are going to take blood that's been oxygenated back to the heart. So this makes pulmonary veins the correct answer. So now that we've taken the mini quiz, let's go ahead and do a few quick review questions to see what you learned today. First, what are some differences between the right and left sides of the heart? Second, why does the systemic circuit have higher resistance than the pulmonary circuit? And lastly, what are the names of each of the heart valves and how many cusps does each of them have? If you can answer these questions, you're well on your way to success. Everybody be sure to have a wonderful day, and I will see you guys in the next one. Hi everyone, Kelton here. Today we're going to be discussing tissue layers of the heart and coronary circulation, so let's jump right in. Like many other organs, the heart is made up of by discrete layers of tissue that differ in their structure and function. So based off of this image of the heart, how many layers would you expect the heart to have? Great, so the heart is typically broken up into three layers: the endocardium, the myocardium, and the pericardium. and each of these has a prefix that's going to indicate something about the location of each layer. So for example, if we take a look at the deepest layer, the endocardium, we see that the prefix endo is going to mean within. So this endo prefix then is really useful for helping us to remember that the endocardium is going to line each of the chambers of the heart and be in direct contact with the blood that's within the heart. Much like the inside of a blood vessel, the endocardium is made up of by cells known as endothelial cells. And if we take a look at these cells, notice that they do a really great job at forming a nice watertight barrier. This is going to make it so that no blood from the heart chamber is allowed to seep through the endocardium and into the myocardium. So in contact with our endocardium, we find our next layer, which is the myocardium here. So if we break down this term myocardium, we find that the term myo means muscle. As the name implies, this muscular layer is going to be the main workhorse of the heart, and it's going to be responsible for pumping blood through and out of the chambers. Recall that this layer is made up of by these cells known as cardiomyocytes. And if you recall, cardiomyocytes is just a fancy term for cardiac muscle cells. The outermost layer is known as the pericardium, and recall that the prefix peri is just going to mean around. This prefix helps us to remember that the pericardium is essentially just a sac that's going to be surrounding the outside of the heart. So it's also important to note that the pericardium has two distinct layers, one on the outside known as the parietal pericardium and one deeper known as the visceral pericardium. And notice there's a small space separating these two layers. The pericardium can be a little bit confusing, so we're going to go into a little bit more detail. So the pericardium is what's known as a serous membrane because it secretes a fluid known as serous fluid. When explaining how this serous membrane surrounds the heart, I always like to use the example of a hand pushing into a balloon. If we think of the pericardium as originally being a balloon and then a hand, the heart, being pushed into this balloon, notice that we're going to form two distinct layers. One layer that's directly attached to the heart, which is the visceral layer, and one layer that's going to not be attached to the heart, which is the parietal layer. Notice that as the heart pushes into the pericardium, we see a small space formed between our two pericardial layers, and this layer is known as our pericardial cavity, or sometimes pericardial space. The serous fluid then is found within the pericardial cavity and is made up of a bunch of proteins that make it ultra slippery. So what do you think the function of the pericardium is in general, and what do you think the function of the serous fluid is? Great, so as we've said before, the pericardium can be thought of as a sac that ends up holding the heart in place. In addition to this then, the serous fluid is going to act as a lubricant that helps reduce the amount of friction the heart's going to experience as it beats. Now that we've talked about the layers of the heart, it's important to address where these layers are going to get their oxygen and nutrients from. So recall that the inner layer, the endocardium, was watertight and it doesn't allow blood to seep through to the myocardium. So where would you expect the myocardium to receive its blood from? Great, so the heart has its own circuit that's responsible for sending oxygen-rich blood out to its tissues and returning the oxygen-poor blood back to the right atrium. This is referred to as the coronary circuit because there's a ring of vessels that sit around the top of the heart that often resemble a crown. So the coronary circuit then is made up of coronary arteries and cardiac veins. So which of these is going to be responsible for delivering oxygen-rich blood to the myocardium? Great, so hopefully you recall that arteries are going to take blood away from the heart and veins are going to deliver blood back towards the heart. So this means coronary arteries are going to deliver blood to the myocardium. Our coronary arteries can be seen here in red on this image, and we can tell these are the coronary arteries because they're coming from our aorta. The cardiac veins are represented in blue, and they're responsible for carrying back the oxygen-poor blood to a structure known as the coronary sinus. The coronary sinus is then where all of the blood will be drained back into the right atrium. Now that we've finished with the lesson, it's time for a mini quiz. Which of the following is not a layer of the heart wall? So it's important to remember that all of the layers we learned today ended in cardium. So the term endocardium, remember, is going to indicate the innermost layer of the heart, the one that lines the chambers. Next then, recall that the myocardium was simply just the muscular layer of the heart. See, it can be a little bit intimidating at first because it has this visceral term, but remember we learned that the pericardium was a sac surrounding the heart, and the visceral layer was the layer that's directly adhered to the heart wall itself. This leaves us with coronary sinus. So recall that the coronary sinus is the small opening in the right atrium where the cardiac veins are going to drain their blood into. And remember, this is not going to be a layer of the heart, making this correct. Now that we've had our mini quiz, let's go ahead and do a few review questions to see what you learned today. First, what are the layers of the heart from deep to superficial? Second, what is the function of the pericardium and the serous fluid? And lastly, describe the pathway of blood from the right atrium to the myocardium and back to the right atrium. If you can answer these questions, you're well on your way to success. Everybody be sure to have a wonderful day, and I will see you in the next one. Hi everyone, Kelton here. Today we are going to be discussing the cardiac conduction cycle, so pause the video, take a look at the outline, and we're going to jump in. To start off with, let's recall that the heart is a muscle, so it's going to have a lot of things in common with the muscles that we would train at the gym. The skeletal muscles that we train at the gym need an action potential in order to make them contract, and the muscle cells from the myocardium are no different. The cells found within the myocardium are going to contract in response to an action potential, so what are these muscle cells in the heart called? And great, so the muscle cells of the heart are known as cardiomyocytes, and if we break this term down, we see that cardio indicates heart, myo indicates muscle, and cytes means cell. One thing that makes these cardiomyocytes different from the other muscle cells in the body is that they have a property known as automaticity. This property of automaticity indicates that cardiomyocytes are capable of generating an action potential with no external nerve telling them to do so. This illustrates a really big difference between the muscle cells of the heart and the muscle cells responsible for moving the body. The muscle cells responsible for moving the body need to be told to contract by a nerve. The heart, on the other hand, doesn't even need to be in a person's body to continue beating. To dive in then to cardiac conduction, we need to first look at the sinoatrial node, which we see here. So the sinoatrial node is sometimes known as the pacemaker of the heart, and this is because it has the greatest automaticity. This means that the SA node is most likely to meet a threshold where it's going to stimulate an action potential that can then ripple throughout the rest of the heart's cells. Although all cardiac cells have this property of automaticity and could technically beat on their own, the SA node is going to fire first and set the rhythm for the rest of the cells in the heart. So based on this image, could you tell me where the SA node is located? And great, so just as this illustration shows, the SA node is located in the right atrium. Even though the heart can function completely independently, it's going to be influenced by the autonomic nervous system. So what do you think the autonomic nervous system, specifically these two branches, are going to do to the heart rate of a heart? Great, so the autonomic nervous system can influence the heart to either speed up its heart rate by utilizing the sympathetic nervous system or slow down its heart rate by utilizing the parasympathetic nervous system. A great way to remember this is para kind of sounds like parents, and my parents always want me to slow down when I'm driving. Recall that the SA node has very quick automaticity, and because of this, the default heart rate is going to be really fast. However, the parasympathetic vagus nerve is going to extend from the medulla oblongata to innervate the SA node. The vagus nerve then inhibits the SA node and slows it down to the normal heart rate of 60 to 100 beats per minute. While talking about heart rates, it's important to address these two terms. First off, tachycardia. Tachycardia is a term that describes a heart rate that is over 100 beats per minute. I like to remember that this is the too fast heartbeat because if you sat on a tach, your heart rate is going to increase. The term for a slow heart rate then is bradycardia, and this implies that you have a heart rate that is under 60 beats per minute. So now we're going to follow the path of cardiac conduction and recall that the SA node is going to be the first to reach its threshold. This is going to send a wave of depolarization through both atria and cause both of them to contract. This will make it so all the blood from the atria ends up filling the ventricles. Once this wave of depolarization reaches the bottom of the right atrium, it's going to encounter a structure known as the atrioventricular node, or AV node. The purpose of the AV node is to add a brief delay between the contraction of the atria and the contraction of the ventricles. If the atria and the ventricles were to contract at the same time, this would cause a lot of problems because the ventricles wouldn't be able to fill completely. After the brief delay, the AV node is going to send an electrical signal to the bundle of His. The bundle of His is located in the interventricular septum. It's called an interventricular septum, which we see here, because it's a wall that separates the two ventricles. The bundle of His then sends an electrical signal to the left and right bundle branches. These bundle branches then pass the signal on to Purkinje fibers. These Purkinje fibers are located within the walls of the ventricles and have the job of making sure the ventricles both contract at the same time. Now that we've talked through cardiac conduction, we need to address two terms, systole and diastole. Although the atria and ventricles both have distinct phases of systole and diastole, the overall cardiac cycle is generally defined by ventricular contraction. This means that the overall cardiac cycle can be said to be in systole if the ventricles are in systole. We can see an example of systole here on the left. So systole is going to occur when the ventricles are ejecting their blood into the arteries they're connected to. So this is when the pressure in the arteries is going to be the greatest. Diastole then is going to occur right after ventricular contraction, and we see that the myocardium in the ventricles is going to be completely relaxed. This phase is going to mark the point when the ventricles are going to be filled, and it also marks the point when the pressure in the arteries is the lowest. Now that we've finished with the lesson, let's go ahead and have a mini quiz. Cells from which of the following have the greatest automaticity? First, we need to recall that automaticity is just a property that means that cells have an ability to generate an action potential on their own. We should also recall that cells with the greatest automaticity are going to end up setting the rhythm for the rest of the heart. We recall then that the SA node is going to act as a pacemaker for the heart because its cells have the greatest automaticity. All of these other structures then are simply going to follow the rhythm set by the SA node. Now that we've had the mini quiz, let's go ahead and do a few review questions to see what you learned today. First, what is automaticity? Second, what is the result of the parasympathetic nervous system acting on the heart? And lastly, if the ventricles are filling, is the heart in systole or diastole? If you can answer these questions, you're well on your way to success. Everybody be sure to have a wonderful day, and I will see you guys in the next one. Hi everybody, Kelton here. Today we are going to be talking about the cardiac cycle, so pause the video, take a look at the outline, and we're going to jump right in. I'm sure that you're all familiar with the sounds that the heart makes, that lub-dub. The crazy part about this is that lub and dub are actually proper medical terms. So what's going to make these heart sounds then? What makes the lub and dub sound? It turns out what's actually creating these sounds is the closing of our valves in the heart, whether they be our atrioventricular valves closing or our semilunar valves closing. To start off, we'll talk about our first sound, lub. So lub is going to occur when we have our atrioventricular valves go from open to suddenly snapping closed. This snapping close is going to create an audible sound that makes the lub. Recall that these atrioventricular valves are going to close in response to ventricular contraction. The second sound then is the dub, and the dub is going to occur when we have our semilunar valves snap closed. Recall that at this time the atria are contracting, the AV valves are open, and the ventricles are being filled. With what we've talked about so far, do you think you could tell me which sounds systole and diastole are going to occur between? Great, so systole is going to occur between the lub and the dub sounds. Recall that systole is simply going to be the point when the ventricles contract and eject their blood into the arteries. Before this can occur, we must have our atrioventricular valve snapped closed, and this generates the lub sound. Once the ventricles begin to relax, the semilunar valves, which were open, are now closed, creating the dub. Rather intuitively, diastole is going to be the exact opposite of systole, and it's going to occur between the dub and the lub sounds. Now we're going to be moving on to signal transduction, and an important question to start with this is what's going to be connecting our cardiomyocytes together. Cardiomyocytes are connected together via intercalated discs. These intercalated discs are made up of by desmosomes and gap junctions. Desmosomes are small proteins that end up connecting our cardiomyocytes. These essentially look like small little stitches that act to stitch the cardiomyocytes together. Gap junctions are protein tunnels that connect the cytoplasm of two neighboring cardiomyocytes. This allows for the transfer of small molecules or ions from one cell to another. This also ensures the action potentials can be propagated from cell to cell. The connection of all the cardiomyocytes via gap junctions is often referred to as cardiac syncytium. Cardiac syncytium is really important to the function of the heart because it allows to act as one unified organ. Lastly, we are going to be talking about electrocardiograms, also known as ECGs or EKGs. As the name implies, an ECG is used to measure electrical activity within a heart, and it ends up creating a little plot like this. To set up an ECG, there are many leads that are placed throughout the body. These leads act as sensors that can sense depolarization when it occurs. Based off of what is known about the lead, like its location, we can then say which region of the heart is depolarizing. We're now going to learn the basic parts of an ECG and then learn what these parts indicate. So first off is our P wave, which we see here. So the P wave is going to indicate that the atria are depolarizing. Next, the slight downline is the Q wave. The Q wave represents depolarization through the interventricular septum. This is going to initiate ventricular depolarization. The tallest up wave on an ECG is known as the R wave, and the R wave is going to represent depolarization through the ventricles. So recall that ventricles are much stronger than atria, therefore it only makes sense that the tallest wave on an ECG is going to be seen during ventricular contraction. Next, we come across this slight up wave. This slight up wave is the S wave, and this is going to indicate the completion of ventricular depolarization. Sometimes the Q, R, and S waves are all read together as one in what we call the QRS complex. The QRS complex just gives a broader view of what's going on during ventricular depolarization. Lastly then, we notice this medium-sized up wave known as a T wave. The T wave is going to represent ventricular repolarization. Here it's important to note that cells are refractory following an action potential. Recall that refractory cells will not experience another action potential until they've completely recovered through a process known as repolarization. You might have noticed that there's no atrial repolarization wave, and this is because atrial repolarization is going to be hidden by the electrically dominant QRS complex. These electrocardiograms are really important clinically because they can indicate to a physician that you might have an underlying heart condition. Now that we've finished with the lesson, it's time for a mini quiz. The dub sound is partly made by which of these valves closing? When looking at this one, there's something that we should notice right away. These are two atrioventricular valves, and we have a right atrioventricular valve as an option. The tricuspid valve and the right atrioventricular valve are the same thing, so A and D already look pretty sketchy. With noting the suspicion of the AV valves, we can then look at B, the pulmonary semilunar valve. Recall that the dub sound is the second sound, and it's made by the audible snapping shut of our pulmonary semilunar valve as well as the aortic semilunar valve. Now that we've taken our quiz, let's go ahead and do a few review questions to see what you learned today. First, between which sounds does systole occur? Second, what is cardiac syncytium? And lastly, label an ECG with all of the parts we discussed today. If you can answer these questions, you're well on your way to success. Everybody be sure to have a wonderful day, and I will see you guys in the next one. Hi everyone, Kelton here. Today we're going to be discussing heart function measurements, so pause the video, take a look at the outline, and we're going to jump right in. The first measurement that we'll be discussing today is heart rate. Heart rate is sometimes referred to as someone's pulse, and it simply indicates how fast or slowly someone's heart is beating. So you're likely pretty familiar with this at this point, so what would you think a typical heart rate would be? Perfect, so hopefully recall that heart rate is measured in beats per minute, also known as BPM, and the typical heart rate falls between 60 to 100 beats per minute. Knowing the normal range then, it's important to address terms like tachycardia and bradycardia. So recall that tachycardia is just going to indicate a heart rate of over 100 beats per minute, and a great way to remember this is if you sit on a tach, it's going to increase your heart rate. Bradycardia, on the other hand then, indicates a heart rate of less than 60 beats per minute. The next measurement we'll discuss then is stroke volume. So stroke volume is going to indicate the amount of blood that is pumped out of the heart at the end of every beat. So this typically refers to the volume of blood that's ejected from the left ventricle, but in most cases it can refer to the right ventricle as well. Recall that this is because the left and right ventricles in a healthy heart will be ejecting the same volume of blood. So, what's a way that you could calculate the stroke volume? A great way to determine stroke volume is to take the amount of blood just before ventricular contraction and subtract that by the amount of blood that's left over after ventricular contraction. This will in theory tell us how much blood was pumped out during that ventricular contraction. If we think about when the ventricles are going to be at their fullest, this is going to be at the end of their filling stage, which remember is diastole. So the volume of blood at the end of diastole is known as our end diastolic volume, also known as EDV. After diastole, systole is going to occur, and recall that during systole, the ventricles are going to contract and eject blood from the heart. At the end of this, there is going to be a little bit of volume left in each of the ventricles, and this is going to be known as our end systolic volume. Once we have our end diastolic volume and our end systolic volume, we can go ahead and use this equation to calculate our stroke volume. And I like to simplify this equation in thinking about it as the end diastolic volume is the amount of blood in the ventricle before contraction. The end systolic volume is being the amount of blood in the ventricle left over after contraction, and the stroke volume being the amount of blood that was ejected from the heart during ventricular contraction. The next measurement we'll be discussing is cardiac output. So cardiac output can be determined by multiplying your heart rate by the stroke volume. So knowing this equation then, what does cardiac output tell us? Great, so as the formula indicates, cardiac output tells us the volume of blood that's pumped by the heart in 1 minute. This is really useful clinically, as having too high or too low of a cardiac output can indicate certain health problems. Next up then is total peripheral resistance, which is sometimes known as TPR. TPR is just going to describe the amount of resistance that blood is going to experience while flowing through the body's vasculature. This is of course because blood is going to be moving through a series of smaller and smaller tubes, and that's going to create a lot of resistance. Total peripheral resistance is affected by both vasoconstriction and vasodilation, so what effect would you expect vasoconstriction to have on TPR? So to answer this question, we first need to recall what vasoconstriction and vasodilation are. So if we start off with a blood vessel of a normal size and it undergoes vasodilation, this means it's going to expand its diameter and become larger. On the other hand, if it undergoes vasoconstriction, it's going to become smaller. In the case of vasoconstriction then, we can think about it as the tube started out this big and there was plenty of room for the blood to flow, and now the tube is much smaller, so there's less room to flow. this is going to mean that there's more pressure within the vessels and it's going to end up increasing your TPR. On the other hand, if we compare a normal vessel with a vasodilated vessel, we see that the vasodilated vessel has a lot more storage space for that blood. This is going to decrease pressure and make it so TPR is going to decrease. The last thing we'll be discussing today is blood pressure, and we've all likely seen blood pressure written like this before. So one important thing to notice right off the bat is that these values are going to be written in a unit of pressure known as millimeters of mercury. This number on the top then represents systolic blood pressure, and this is going to be when the pressure in your arteries is at its highest. So this is going to occur in systole when your ventricles are contracting. The bottom number then represents your diastolic blood pressure, and this is going to represent the lowest pressure in your arteries, and this is going to occur when the heart is in diastole and relaxing. Seeing blood pressures written like this can be a little bit misleading because these two values aren't equal in their importance. Diastole lasts for longer than systole, thus diastolic blood pressure has a little bit more bearing when it comes to your health. So what's a better, more effective way to get an accurate blood pressure reading? And great, so you may be familiar with mean arterial pressure, and this is just going to indicate the average arterial pressure during one complete cardiac cycle. Because of this, it's considered to be a much better indicator of blood pressure than systolic or diastolic blood pressure. To determine mean arterial pressure, you simply multiply your cardiac output by your total peripheral resistance. But with what we've learned today, is there another formula you could use to find mean arterial pressure? Great, so hopefully you recall that cardiac output is equal to stroke volume multiplied by heart rate. So if we don't have cardiac output, we can just plug in these two measurements in its place. Another important part of this last formula is that it shows us if we increase stroke volume, heart rate, or total peripheral resistance, it's going to end up increasing mean arterial pressure as well. In order to measure systolic and diastolic blood pressure, many clinicians use a technique known as auscultation. This technique utilizes a sphygmomanometer, which is a cuff that wraps around your arm and has a little gauge for reading pressure, and then a stethoscope to listen for the blood to flow through the artery. Although many clinics are starting to move away from this because there's a lot of automatic versions of this now. Now that we've finished with our lesson, let's go ahead and do a mini quiz. Which of the following can be used to find mean arterial pressure? So remember that we had two different equations to find mean arterial pressure. The first of which was that mean arterial pressure was equal to cardiac output multiplied by total peripheral resistance. Another way we found mean arterial pressure was by using pretty much the same equation but swapping our cardiac output with stroke volume multiplied by heart rate. Now we can go ahead and compare this with the answers, and as we see, D is going to be our correct answer. Now that we've had our mini quiz, let's go ahead and do a few review questions to see what you learned today. First, what is systolic blood pressure? Second, what is the range of a healthy heart rate? And lastly, what is the stroke volume and how can it be calculated? If you can answer these questions, you're well on your way to success. Everybody be sure to have a wonderful day, and I will see you in the next one. Hi everybody, Kelton here. Today we're going to be discussing blood vessels, so pause the video, take a look at the outline, and we'll jump right in. To start off, we're going to be discussing blood vessels in general, and you're probably familiar with blood vessels to some extent, and many of you have likely seen a diagram like this before. So, with what you know now, how would you describe what a blood vessel is, and what are some different examples of blood vessels? Perfect. So the basic definition of a blood vessel is a hollow tube that's used to transport blood from one area of the body to another. Two really common examples of blood vessels are arteries and veins. We'll learn about the others later on. So another thing to notice is that just like the heart, these blood vessels also have distinct tissue layers. Before we talk about the different tissue layers of this vessel, it's important to note that this hollow portion, or this central pore of a blood vessel, is known as the lumen of a blood vessel. Lumen is a really common anatomical term that's used to describe the space within a tubular structure. So that can be the lumen of a blood vessel that's filled with blood, or the lumen of your intestine that's going to be filled with your digesting food. Moving on then to the actual layers of a blood vessel, we see the term tunica is going to be referring to each one of these layers. So we know that here there's going to be 1, 2, 3 distinct tunics of a blood vessel. The layer that makes contact with the lumen of a blood vessel is going to be the thinnest layer, and it's known as the tunica intima. A great way to remember this is that the tunica intimate is going to be intimate with or touching the lumen of a blood vessel. So tunica intima is made up by a single layer of epithelial cells. Do you recall the name of these epithelial cells here? And as a hint, these are going to be the same type of cells that you find within the endocardium of the heart. And great, so tunica intima is going to be made up of by a single layer of endothelial cells, and they have the job of reducing the amount of friction blood is going to experience while flowing through a vessel. The layer superficial to tunica intima is known as tunica media. This is going to be our meaty layer, and it's going to be made up of by smooth muscle cells. I always like to remember that this M stands for muscle. This smooth muscle can change the diameter of a blood vessel, and by doing so, manipulate how much blood is going to be allowed to flow through that vessel. If we have a blood vessel that starts off at a normal diameter here, and then the tunica media suddenly undergoes a contraction of its smooth muscle, this is known as vasoconstriction. And notice that the diameter of a blood vessel has decreased greatly, allowing less blood to flow through. On the other hand, though, if the smooth muscle of tunica media relaxes, this is known as vasodilation, and now more blood can flow through that vessel. The most superficial tunic is known as the tunica externa, and it's really aptly named because it's the most external of all the tunics. This layer is made up of by connective tissue and collagen proteins that help to protect the blood vessel, as well as a protein known as elastin. Elastin acts much like an elastic does and helps a blood vessel return back to its original size after being stretched. Tunica externa is also the place where nerves are going to enter into a blood vessel and eventually communicate with the smooth muscle within tunica media and influence vasoconstriction and vasodilation. Now that we've learned all of the layers of a blood vessel, let's go ahead and do a quick quiz to see if you've got it down. So label this image and also try to see if you can point out some differences between arteries and veins. Great, so right off the bat, we should notice that the layering of arteries and veins is going to be the same. We start out with a lumen in the center, we have tunica intima next, after that we have our tunica media, and finally we have tunica externa. Some differences you may have noticed are that veins utilize valves while arteries do not, and arteries have a much thicker tunica media than veins do. We'll be talking about this more later, but it's important to know that arteries are going to be pumping blood away from the heart and towards a capillary bed. They are also going to be under much higher pressure than veins. Now we'll be talking about the first two types of vessels we'd encounter after leaving the heart. So, what do you think arteries and arterioles are going to have in common, and how might they be different by looking at this image? Great, so arteries and arterioles are both going to carry blood away from the heart, and they're both highly elastic. Remember, the property of a vessel being elastic is that they're able to be stretched like this and return back to their normal state fairly easily. Based off of what we've talked about so far, why do you think arteries and arterioles are so elastic? And great, a thick tunica media is going to make a blood vessel much more elastic. In addition to this, having a high amount of elastin protein is going to make a blood vessel much more elastic as well. Recall that arteries is where pressure is going to be the highest, so arteries having a high degree of elasticity is very beneficial because this allows for that high pressure to be moderated, and what that means is that high pressure can be evened and spread out along the entire vessel. Some of the main differences between arteries and arterioles is that arterioles are much smaller and they end up having a much lower pressure than the pressure in arteries. Arterioles is going to be where blood pressure experiences the greatest drop, and we also see that arterioles are commonly referred to as resistance vessels, and this is going to be because there's a large amount of resistance that blood is going to have to face while flowing through one of these arterioles. Arteries and arterioles are both going to experience vasoconstriction and vasodilation, but the effect of vasoconstriction and vasodilation is going to be a little bit more important within our arterial system. So, when do you think an arteriole might undergo vasoconstriction? And great, so recall that vasoconstriction is going to decrease the amount of blood that's able to flow through a vessel. So if arterioles are going to undergo vasoconstriction, that means that not as much blood is needed or wanted in a certain area. So to think of a specific example of that, recall that we are homeothermic endotherms, meaning that we maintain a constant body temperature. So how might vasoconstriction and vasodilation of arterioles assist us in this? On a day like a cold day, we lose a lot of heat to the environment from our integument. In order to combat this, the arterioles within the integument can either vasodilate or vasoconstrict. If it's a really cold day outside, the vessels in our integument are going to undergo vasoconstriction. And this is going to be because we want less blood to be exposed to that cold environment. Instead, we're going to keep our core temperature high while letting our extremities get a little bit cooler. If it's a warmer day outside, the opposite is going to occur and the vessels within the integument are going to undergo vasodilation. This is going to mean that more of our blood is going to be exposed to that cool environment and helps keep our body temperature relatively low. Now that we've finished with the lesson, let's go ahead and do a mini quiz. Endothelium is found in which of these layers? To start off with A, we know that this prefix myo is going to indicate this is a muscular layer, specifically a muscular layer of the heart, so this is incorrect. Next we have tunica externa, but recall tunica externa is the externalmost layer, and it's made up of by connective tissue, collagen proteins, and elastin proteins. For C, hopefully recall that tunica media is going to stand for the muscular layer of a blood vessel. This leaves us with the correct answer choice D. Recall that tunica intima is going to be made up of by a single layer of epithelial cells known as endothelium. Now that we've had our mini quiz, let's go ahead and do a few review questions to see what you learned today. First, list each of the layers of a blood vessel, as well as characteristics of each. Second, what are arterioles? And lastly, how does vasoconstriction assist in thermoregulation? If you can answer these questions, you're well on your way to success. Everybody be sure to have a wonderful day, and I will see you guys in the next one. Hi everyone, Kelton here. Today we are going to be discussing capillaries, so pause the video, take a look at the outline, and we're going to jump right in. To start off, we need to address what a capillary is, and by now we've heard this term quite a bit, so how would you describe what a capillary is to a friend? Go ahead and use this image to help explain it. Great, so capillaries are the smallest blood vessels based off of diameter, and notice that they're so small in fact that red blood cells have to pass through them single file. Capillaries can be seen on this image here, and notice that they're going to end up linking our arterioles and our venules together. These capillary beds are going to be a key site for nutrient exchange. Notice that this image shows that oxygen is giving off to the surrounded tissues from the oxygen-rich portion of the capillary bed, and then the oxygen-poor blood is returned back to the heart. In addition to this, capillaries are also where nutrients like sugar or amino acids are given to the surrounding tissues. These capillary beds also end up taking up waste from the surrounding tissues like carbon dioxide and sending it back to the lungs where it can be expelled. As this picture demonstrates, capillaries are really simple because they only utilize one cellular layer, our tunica intima, which if you recall is just our endothelium. Recall that this is different from other vessels, as other vessels like arteries and veins typically utilize 3 tunics. Capillaries do, however, have a non-cellular layer known as the basement membrane that acts to give the capillary a little bit more structural support. So why do you think it is that our capillaries are so small in diameter and have such thin walls? And great, so hopefully you recall that capillaries are so small and thin because oxygen is distributed to the tissue by simple diffusion. Recall that how thick a membrane is and the amount of surface area are 2 factors that affect how quickly simple diffusion can occur. If we compare the wall of an artery with the wall of a capillary, we notice that the wall of the capillary is going to be much thinner, and this allows for oxygen from our red blood cell to be distributed to the tissues much more efficiently than in the artery where there's so many more layers the oxygen would have to diffuse through. Next, if we compare the diameter of an artery with the diameter of a capillary, notice that in a capillary, all of the red blood cells are forced to travel one by one single file, and all of them are making contact with our tunica intima. If we look at the artery then, there's a lot of red blood cells in the middle here that are never even going to get the chance to do gas exchange because they never would touch the tunica intima. Instead, having all of the blood from these arteries delivered into thousands of capillaries, this is going to maximize the amount of surface area and thus maximize the ability for nutrient and gas exchange. While many materials can simply diffuse into or out of a capillary, some materials are just simply too big or too polar to do so, and that's where these fenestra or pores come into play. While we had learned earlier that tunica intima was typically made up of by endothelial cells that formed a watertight barrier like we see here, this isn't always the case. In certain types of capillaries, they utilize fenestrations or fenestra, which are little holes that allow larger nutrients to pass into and out of the capillaries. In addition to this, there are also sinusoidal capillaries that have even larger holes. This allows for even larger materials to enter into and out of a capillary. Now we're going to discuss how fluid, specifically blood plasma that has lots of proteins and other nutrients in it, is capable of moving from inside a capillary to the interstitial fluid that's surrounding the tissues. So if we think of ourselves as a protein that's within the plasma, we're going to be flowing through an arteriole and make our way into a capillary here. Inside of this capillary, eventually we might be taken out into our interstitial fluid while the red blood cells are going to continue until they reach the venule. The first force that we are going to address is capillary hydrostatic pressure, and this is just going to be a force exerted from the fluid pressure within a capillary that's going to end up driving our plasma out. From here, the plasma can then nourish the surrounding tissue, but it's important to note that a force known as interstitial fluid hydrostatic pressure is going to be opposing this. These arrows don't give a good indicator of which force is going to be greater, so based off your knowledge of blood flow, is our capillary hydrostatic pressure or our interstitial fluid hydrostatic pressure going to be greater? Great, so capillary hydrostatic pressure is going to be the greater of these two forces, and we can conclude that this is pretty reasonable because on the arterial side, we need more blood plasma to be exiting our capillary than to be entering it. If more entered than exited, our tissues would never get enough nutrients. Moving on then to our venial side of a capillary bed, we see that the dominant force is our plasma osmotic pressure. So this plasma osmotic pressure is going to be pulling fluid from the interstitial fluid back into the capillary. So what's osmotic pressure and why is this occurring? Great, so hopefully you recalled that when we have two solutions that have different concentrations and they're separated by a semi-permeable membrane, they have different osmotic pressures. The one with more solutes has a higher osmotic pressure than the one with fewer solutes, which has a lower osmotic pressure. With this, it's important to know that water is going to flow from areas of low osmotic pressure towards areas of higher osmotic pressure, and that's exactly how plasma is going to make its way back into the capillary. At the distal end of our capillary, there's going to be a really high concentration of protein as compared to our interstitial fluid. This means that the fluid from here is going to end up flowing back into the capillary with a lot of the substances that are dissolved in it. You may have noticed that there's an opposing force to the plasma osmotic pressure as well, and it's known as our interstitial fluid osmotic pressure. Now this is generally the weaker of the two forces, and it has the main goal of making sure not too much of the fluid goes back into the capillary. This may seem a little bit complicated at first, but we can summarize it by saying that on the arterial side of a capillary, there's going to be a net movement of fluid out of a capillary due to our hydrostatic capillary pressure. And on the venial side, we see the osmotic pressure is going to pull fluid back into the capillary. Now that we've finished our lesson, let's go ahead and take a mini quiz. Which of these is never found in a capillary? So if we look at A and B, these two are both going to go together because remember, tunica intima is always made up of by our endothelium. Recall that a capillary has a tunica intima as its only layer, so these are both incorrect. For answer C, recall that fenestra are simply small pores that allow for larger nutrients to enter and exit capillaries more efficiently, so this is incorrect. And now we come to the last answer, tunica media. So recall that tunica media is the layer of a blood vessel that's going to be made up of by smooth muscle. Capillaries do not have this as they only utilize tunica intima. Now that we've finished with our mini quiz, let's go ahead and do a few review questions to see what you learned. First, describe the structure of a capillary. Second, why is a capillary suited for delivering nutrients to tissues? And lastly, describe the flow of fluid from a capillary to tissues, then back to the capillary. And be sure to talk about why the fluid moves the way it does. If you can answer these questions, you're well on your way to success. Everybody be sure to have a wonderful day, and I will see you in the next one. Hi everyone, Kelton here. Today we're going to be discussing the venous system, so pause the video, take a look at the outline, and we're going to jump right in. The venous system is made up of by vessels known as venules and veins. If we take a look at this image, we see that our venules are going to be receiving blood from our capillaries. From here, the venules then port their blood into a much larger vein. Veins are much wider than arteries, meaning they store much more blood. At any moment in time, most of your body's blood is actually found within the venous system. Because of this, veins are known as capacitance vessels, meaning they essentially act as a storage unit for our blood. So why do you think veins are able to hold so much more blood than arteries? Great, so if we compare the smooth muscle layer of an artery with that of a vein, notice that the smooth muscle layer of an artery is much thicker than that of a vein. This means that there's much more space for blood to be stored in a vein than in an artery. Because veins are under low pressure as compared to arteries, they really don't need a thick, smooth muscle layer. So, can you recall the proper name for the smooth muscle layer of a blood vessel? Perfect, so recall that the muscular layer of a blood vessel is the tunica media, with the M standing for muscle. So to get to a vein, we have to travel through a series of arteries, and notice that in all of these arteries, there's going to be really high pressure. If we move on then to our arterioles, notice there's a very steep drop in pressure as we approach our capillaries. After passing the capillaries then, we move into our venous system where we see that the pressure is even lower. So with these low pressures and really thin tunica media, how is blood going to make its way back to the heart, and what ensures that the blood isn't going to flow backwards? To tackle this question, let's first address why when we're standing up, blood in our legs doesn't start to pool at the bottom of our feet. Gravity is constantly pulling the blood in our legs back down towards our feet, and to counteract the blood from flowing backwards, we see that medium-sized venules and all veins utilize things that are known as valves. We can think of these valves as having a similar function to that of the valves in the heart. They prevent backflow. If we look at this diagram of a valve, we see that whenever blood's flowing this way, it can easily push open those two flaps to move in the correct direction. On the other hand, though, the structure of the valve makes it so that if blood tries to go back this way, it ends up closing off the valve and doesn't allow blood to head back down towards our feet. So while these valves are really useful at preventing backflow, they don't really do anything to help propel the blood towards the heart. So how do you think that blood's going to get pushed back towards the heart? Great, so the venous system utilizes two different pumps to help push blood back towards the heart. The first of which is the skeletal muscle pump. As the name implies, this is going to be a pump that utilizes the skeletal muscles to push blood. So if we think of this as a vein that's found near the gastrocnemius muscle, there's always going to be gravity pulling the blood back down towards your feet. Thankfully though, your valves are going to prevent this blood from flowing too far back. But let's say you start to do some calf raises. As the gastrocnemius muscle ends up contracting, it's going to squeeze on this vein and push our blood back up towards the heart. This image does a really great job at illustrating that even though some of the blood is pushed backwards, the one-way valve is going to prevent it from flowing too far back, so we end up with a net movement of blood going straight towards the heart. Because the venous system is so dependent upon the skeletal muscle pump, a lot of people who are paralyzed and don't have the ability to move their legs end up experiencing a lot of complications like edema and deep vein thrombosis. The other pump used to get blood back towards the heart is the respiratory pump, or sometimes called the abdominothoracic pump. It's called the abdominothoracic pump because it utilizes our thoracic cavity's pressure, which is above the diaphragm, and our abdominal cavity's pressure, which is below the diaphragm. If we start from a position where we've just fully exhaled and begin to inhale, what's going to occur is our diaphragm is going to contract and it's going to pull the pleura of the lungs downwards. So this is obviously going to create a little bit of pressure in the abdominal cavity where it's going to start to squish all of your guts together. This increased abdominal pressure is going to end up squeezing on any of your veins in the abdomen and helping to push that blood back in the correct direction towards the heart. Along with an increase in abdominal pressure, we also see that during inhalation, there's going to be a decrease in thoracic pressure. This is useful for two reasons. One, a decrease in pressure is going to cause our air to flow into the lungs. And two, there's going to be a negative pressure here that's going to cause a vegetative force on the vena cava that's going to act to help suck blood back into the right atrium. Now that we've finished with the lesson, let's go ahead and do a mini quiz. Which of the following is a characteristic of a vein? So with A, right off the bat, we know that veins utilize a thin tunica media and not a thick tunica media, making A incorrect. B looks pretty good, so we'll come back to that one later. C says fenestra, and remember, fenestrations are just small holes that are found within capillaries, not veins. And lastly, D says resistance vessel. So remember, arterioles, where there was the greatest decrease in pressure, are going to be our resistance vessel. Veins, on the other hand, are capacitance vessels. So this confirms that B is the correct answer. Remember that medium-sized to large-sized venules utilize valves, as well as all veins. Now that we've had our mini quiz, let's go ahead and do a few review questions to see what you learned today. First, describe the structure of a vein. Second, how does venous blood return to the heart? And lastly, which vessels have the lowest pressure? If you can answer these, you're well on your way to success. Everybody be sure to have a wonderful day, and I will see you in the next one. Hi everyone, Kelton here. Today we're going to be talking about portal systems, so pause the video, take a look at the outline, and we're going to jump right in. So to start off, let's ask the question of what is a portal vein system, and how does it differ from another circuit like, say, the pulmonary circuit? Great, so portal systems are going to occur when two capillary beds are connected to each other via a portal vein. So if we take a look at this diagram on the left, if we're a red blood cell flowing through arterial circulation, we're eventually going to make it through an arteriole. From this arteriole then, we're dumped into our first capillary bed. After this, we enter a series of venules and veins that make up a portal vein because they're porting us to a second capillary bed. In the image on the right, we can see a practical example of this with the blood from the capillaries in the stomach and intestines entering into a hepatic portal vein and then entering into the liver where they meet a second set of capillaries. These portal systems are really important to us, but at a first glance, it looks really inefficient because we're sending oxygen-poor blood to a second set of capillaries. So what's the function of these portal systems? Great, so portal systems are really important because they allow for the shunting of concentrated blood from one area to the other. This is really useful for two main reasons. First, if we think about all the stuff that we eat for lunch, if we sent that directly back into the systemic circuit, it could end up harming things like the brain. A portal system allows for this blood to be sent somewhere first and processed and made safe for the rest of the body. The second reason portal systems are so useful to us is that the hypothalamus is able to send blood that's really concentrated with hormones directly to the pituitary gland rather than having those hormones go through the entire systemic circuit. As we touched on before, having really concentrated blood is dangerous to certain organs like the brain. Now, during digestion, we see that organs like the small intestine are going to be absorbing whatever nutrients and whatever toxins we had for lunch that day. If this was all allowed to simply flow back towards the heart and the brain, it'd be delivering a full load of toxins that way. But the hepatic portal system allows us to avoid this. The main goal of the hepatic portal system is to take concentrated blood from the digestive organs and a few other organs like the spleen and send them towards the liver where it can be processed and made safe for the rest of the body. On the right, we see a really basic diagram of how the hepatic portal system works, with blood from the stomach and the intestines being ported through a vein called the hepatic portal vein and eventually making their way into the liver. On the left, we see a little bit more complicated but very famous anatomical drawing from Gray's Anatomy that illustrates all of these veins that are eventually going to flow into the hepatic portal vein before they reach the liver. Our other portal system is a little bit lesser known, and it's called the hypophyseal portal system. The purpose of this system is to shunt blood from our hypothalamus to our anterior pituitary gland, which is sometimes known as your adenohypophysis, meaning glandular part of the pituitary gland, and this is where the portal system gets its name of hypophyseal. Sometimes you'll even hear this portal system referred to as the hypothalamo-hypophyseal portal system, and that's simply because it's connecting our hypothalamus with the adenohypophysis. As a quick refresher, recall that the hypothalamus tells the anterior pituitary gland what to do by sending hormones its way. As this image demonstrates, the hypothalamus and pituitary gland are really close together, but if a portal system wasn't used here, blood from the hypothalamus containing hormones would have to go to the heart and eventually hopefully make its way back to the anterior pituitary gland. This would cause quite a few issues because it would take a lot longer for the hypothalamus to communicate with the pituitary gland, and it would take a lot more hormones to do so. Luckily for us though, we do have a hypophyseal portal system, and this allows for really rapid communication as hormones from the hypothalamus can be released into this capillary, and from there this blood is ported via hypophyseal portal veins into our capillary that surrounds the anterior pituitary gland. Now that we've finished with our lesson, let's go ahead and do a mini quiz. All of the following are part of a portal system except, to answer this question, it's important to note that portal systems are often referred to as venous systems because they include two capillary beds and the veins and venules that connect the two. If we think about this in terms of the hepatic portal system, we see that hepatic capillaries are included in the portal system as they are the final destination of blood from our small intestine. Similarly, duodenal capillaries are also involved because they're going to give their blood to the hepatic portal vein. Looking at D, the term hepatic portal vein already implies it's part of a portal system, and this leaves us with C as the correct answer. Hepatic arteries are not part of a portal system because they're not involved from shunting blood from one capillary to another. Now that we've finished our quiz, let's go ahead and do a few review questions to see what you learned today. First, what is a portal system and what is its purpose? Second, where are the two portal systems in your body? And lastly, why are portal systems considered to be venous systems? If you can answer these questions, you're well on your way to success. Everybody be sure to have a wonderful day, and I will see you in the next one. Hi everyone, Kelton here. Today we are going to be talking about the blood clotting cascade, so let's jump in. To start off, let's acknowledge that blood vessels are going to get damaged. There's nothing that we can do to avoid that, whether it's a fall and we scrape ourselves, or whether we have a vessel that's in the subconjunctiva pop. Now, losing a little bit of blood like this isn't typically going to be a problem, but what's going to make it so that we stop bleeding eventually? Because if we keep bleeding forever, we're going to end up like this flat tire and completely run out of blood. So what mechanisms does our body have that prevents this from happening? Great, so as the title of the video suggests, our body utilizes a blood clotting cascade to fix our blood vessels, and this works pretty similarly to how we would fix a flat tire. If there's a hole in a tire, the first thing that we need to do is put a plug in that tire to make sure that no more air escapes. Our blood vessels are really similar. If we have a hole in a blood vessel, we need to plug up that hole so that no more blood escapes. Now it's going to be a little bit more complex, but it's basically the same idea. There's a hole and we're going to patch it. The blood clotting cascade is a positive feedback mechanism, which, if you remember, implies that the product of the reaction is going to make it so the reaction takes place more. So, what do you think the main players are going to be that's going to start this cascade? Great, so the first step in this cascade is that the tissue is going to become damaged, and when the tissue is damaged, it's going to release signals that are going to end up attracting platelets. Platelets are just cell fragments that are known to be involved with helping form clots. Now, where we really see the positive feedback mechanism part of this is that the platelets that have arrived will release more signals to attract more platelets to the scene, and they're going to continue to do this until a clot has been formed. Overall, this is the basic part of the blood clotting cascade, but now we're going to break it down into more accurate and smaller parts. So as we just said, the clotting cascade is initiated by tissue damage to a blood vessel. This can be in the form of a cut or a tear of a blood vessel. Recall that there's a protein known as collagen that's typically found in the walls of most blood vessels, and this tear or cut is going to end up exposing that collagen to the blood. In the next step, we see that the platelets that are floating around in the bloodstream end up interacting with this collagen and become activated. This activation causes the platelets to begin to adhere and aggregate at the damaged area of our blood vessel, and in doing so, they end up creating a plug known as the platelet plug. Next, our activated platelets begin to release something called thromboplastin, which is also known as tissue factor. Here we can see that thromboplastin has the important role of converting prothrombin to thrombin. As a review, recall that many enzymes have both an active and an inactive form. Typically, we see that enzymes in an inactive form have a pro in front of their name. So for example, we see that prothrombin is the inactive form of the enzyme thrombin. Now on to our last step. So the thrombin that we made in step 3 is really important because it has the role of converting fibrinogen into fibrin. Fibrinogen is simply just the precursor to fibrin, and thrombin acts to cut up that fibrinogen into fibrin. At this point, fibrin is going to begin polymerizing with other fibrin strands, and after it's done so, it's going to attach itself to the platelet plug and form our hemostatic plug, which is also just known as a blood clot. So as you can see, this isn't quite as simple as just plugging up a tire, but if we remember it as these 4 simple steps, it's not too bad. The last thing we are going to discuss is the importance of vitamin K to the clotting cascade. Vitamin K is important because it's required for making many clotting factors, including prothrombin. We typically can get vitamin K through our diet, but we actually get most of our vitamin K through a symbiotic relationship with bacteria in our gut. So, knowing this then, who do you think might suffer from problems of vitamin K deficiency? Great, so you may have heard before that newborns have a pretty high chance of having vitamin K deficiency related bleeding. This is because there's only a small amount of vitamin K stored in the newborn's body at the time of birth because not a lot of vitamin K is passed from the mother to the newborn. In addition to this, at the time of birth, babies don't have that good bacteria that's responsible for producing vitamin K. And if all these factors weren't enough, to make things a little bit harder for Earth's newest member, there's not a lot of vitamin K in breast milk either. So because of all this, typically newborns are given a shot of vitamin K that's going to last them for long enough for the good bacteria to go populate the large intestine. Now that we've finished with the lesson, let's go ahead and do a mini quiz. Activated platelets release which of the following? So starting with A, recall that vitamin K we get from our diet or from our symbiotic bacteria in our gut, so this is incorrect. Answer B, collagen. Remember that collagen is actually what activates the platelets, not what they release. For answer C, remember that thrombin doesn't actually interact with the platelets directly and instead interacts with fibrinogen. This leaves us with the correct answer, D, thromboplastin. Remember, thromboplastin is released by activated platelets and it has the job of converting prothrombin into thrombin. Now that we've had our mini quiz, let's go ahead and do a few review questions to see what you learned today. First, describe the steps of how a hemostatic plug forms. Second, what is the role of thromboplastin in the clotting cascade? And lastly, what is the precursor to fibrin, and what is responsible for its conversion to fibrin? If you can answer these questions, you're well on your way to success. Everybody be sure to have a wonderful day, and I will see you in the next one. Hi everybody, Kelton here. Today we're going to be discussing blood. So pause the video, take a look at the outline, and we're going to jump right in. You're likely already pretty familiar with what blood is. We know that it's found in animals with a closed circulatory system. We know it's a type of connective tissue. We know that it looks red. But what's actually in blood? So if we take a vial of blood from someone and we put it into a centrifuge and spin it down, we find that it separates into 3 main parts. We have plasma, which makes up the most of the volume. We have erythrocytes, which make up the second most of the volume. And the smallest are our leukocytes and our platelets. And we're going to talk about each of these in a little bit more detail. Most of our blood volume is made up of by this gross yellowish liquid known as plasma. So plasma is basically just water, but it has a lot of proteins, nutrients, and even hormones in it. Many of you likely know someone who has donated plasma before, which is a process where you go into a plasma center, they stick you with a needle, they take your blood out, they separate your red blood cells from the plasma, and then they give you your red blood cells back while keeping the plasma. This is really useful clinically because your plasma or your proteins in the plasma can be given to someone who doesn't have the ability to make that protein on their own. A very small amount of our blood volume is made up of by leukocytes and platelets. So leukocytes, as the name implies, are just white blood cells. Here we see a few different examples of white blood cells, and notice that they all possess a nucleus. So what's a function of a white blood cell? And great, so white blood cells are often thought of as the backbone to the immune system, and you're going to be learning about the specific functions of white blood cells in a later video. Platelets, which are also known as thrombocytes, are cell fragments that do not contain a nucleus, and because of this, they're known as being anucleate. Platelets originate from cells known as megakaryocytes. These megakaryocytes are quite large cells, as you can see, and they're found within the bone marrow. These megakaryocytes essentially cut off small pieces of themselves, and these small pieces end up becoming platelets. So the platelets then can enter into the bloodstream and go be involved with the clotting cascade. Now on to our last component of blood, erythrocytes. So based off of this name, what do you think erythrocytes are? And great, so if we break down this name, we see that cytes means cells and eryth means red. So these are our red blood cells. You probably already know that the main job of red blood cells is to deliver oxygen throughout the body. Because of this, they have a cell that's very well suited for this. The shape of this blood cell is biconcave, and the reason for this is that it maximizes the amount of surface area to the amount of volume. This makes gas exchange more efficient. In addition to this, if we look at this microscope slide, notice that these red blood cells do not have a nucleus. Really, they don't seem to have very much of anything in their cytoplasm. The reason for this is that if they don't have as much stuff in their cytoplasm, they're cheaper to make, and there's more space to store oxygen. Another key attribute of a red blood cell is that it's very flexible, which allows it to squeeze through capillaries. So how are red blood cells able to carry oxygen so well? And the answer is found in this little molecule known as hemoglobin. A single red blood cell contains millions of molecules of hemoglobin. Hemoglobin is a quaternary protein, meaning it's a protein that's composed of by multiple different protein subunits. Here we can actually see an example of what hemoglobin looks like, and notice that there are 4 subunits for a single hemoglobin molecule. Each one of these subunits is strongly associated with a non-protein heme group, which we see here. Notice that this heme group contains one iron molecule, and this iron molecule is capable of binding to a molecule of oxygen. This means that each molecule of hemoglobin is capable of binding to 4 molecules of oxygen. Or if you want to think about it in a different way, one molecule of hemoglobin can carry 8 oxygen atoms. Another key trait of red blood cells is that they have antigens on their surface. These antigens are going to be made up of by sugars and proteins. So what's the purpose of these antigens? Great, so these surface antigens are really important because they're what gives us our blood types. Clinically, this is very important because if we give someone a blood transfusion of a blood type that they make antibodies against, it's going to cause a lot of problems, and we're going to dive into that now. There are 4 main blood types based off of A and B surface antigens, and they're all named really well too. So if we start off with type A, we see that type A is going to utilize an A antigen on its surface. Similarly, if we look at a type B blood cell, it's going to have B antigens on the cell surface. As you might have guessed then, type AB red blood cells utilize both A and B antigens on their cell surface. So knowing this, what type of antigens would you expect to be on the surface of a type O red blood cell? So this one may have been a little bit confusing because type O red blood cells do not have a type O antigen. Instead, type O red blood cells simply do not have A or B antigens. So I like to think of this O as being a 0. Next, we're going to take a look at antibodies. And remember, antibodies are just our body's way of protecting ourselves against foreign invaders. Recall that antibodies are going to bind to an antigen. So if we have type A antigens in our blood, we don't want to have type A antibodies. The type A antibodies would bind to the type A antigens and cause a lot of problems. Instead, someone with type A blood is going to make antibodies to type B blood. Moving on then to a person with type B blood, they're going to generate antibodies that attack type A red blood cells. Type AB then doesn't make either of those two antibodies, and that's because it possesses both of those antigens. In other words, it doesn't see type A antigens or type B antigens as something foreign. Lastly, if we look at our type O red blood cells, we see that they make both antibodies against the A antigen and the B antigen. And this is because type O red blood cells don't make either of those two antigens, so it sees them as foreign. In addition to A and B cell surface antigens, red blood cells also sometimes have a protein known as rhesus factor, or Rh. This system is a bit more simple in that you either have rhesus factor or you don't have rhesus factor. If a person has rhesus factor on the surface of their red blood cells, they're known as Rh positive, and a person who does not have it is Rh negative. As you might have guessed, if a person is Rh positive, they're unable to donate to a person who's Rh negative, and this is simply because an Rh positive red blood cell is going to have those rhesus factor proteins on the cell surface. When we combine these two systems together, we see that we have a total of 8 different blood types that you can be. When talking about blood transfusions, there's a few terms that are useful to know. The first of which is agglutination, and this is something that happens when you give someone the wrong type of blood. For example, let's say I'm type O. That means that I make antibodies against type A and type B blood. If they gave me a transfusion of type B blood, those antibodies would then start to agglutinate and stick together those erythrocytes. This is going to cause a lot of issues as the immune response isn't going to just harm the donor's blood, it's also going to harm me as well. Two other terms that come up quite a bit when discussing blood transfusions are the terms universal donor and universal acceptor. So what do you think these two terms mean, and what types of blood do you think each of those would be? And great, so as the name implies, a universal donor is a person who can donate blood to any other blood type. This means that this person must have no antigens on the surface of their red blood cells. That includes they don't have rhesus factor and they don't have A or B antigens, and that would make this person type O negative. On the other hand, a universal acceptor is a person who can receive blood from any of the other blood types. Type AB positive is perfect for this because they have rhesus factor on the cell surface and they also have the A and B antigens. So now that we've finished with the lesson, let's go ahead and do a mini quiz. A person with only A antigens and rhesus factor can donate to which of the following blood types? So right from the start, we should recognize that this is a person who only has A antigens and rhesus factor, meaning that this person has type A positive blood. From here, remember that anyone who is Rh positive can only donate to others who are Rh positive, so this gets rid of A as well as C. Next, recall that a person with type A antigens can only donate to another person with type A antigens. So if we look at D, we see this is incorrect because this guy only has type B antigens. This leaves us with answer B, which is correct because you can donate to someone with your same blood type. Now that we've taken our mini quiz, let's go ahead and do a few review questions to see what you learned today. First, what are the 3 components of blood? Second, describe the components of an erythrocyte. And lastly, which blood types make antibodies against type B? If you can answer these questions, you're well on your way to success. Everybody be sure to have a wonderful day, and I will see you in the next one. Hi everyone, Kelton here. Today we are going to be discussing fetal circulation, so pause the video, take a look at the outline, and we're going to jump right in. So to start off, a fetus is going to begin development by developing in a fluid environment of the mother's uterus. So how is this fetus going to end up getting the oxygen that it needs to develop? Great, so the way that a fetus ends up getting the oxygen that it needs to grow and develop is through its mother. The fetal circulatory system has all these adaptations that allow the fetus to essentially pull oxygen and nutrients from the mother and use them to grow. Because fetal circulation is a little bit complex, I like to envision myself as being a red blood cell that's going to follow either one of 2 paths. The first path is the pathway of oxygen-rich blood from the placenta to the fetus, and the second path is the path of oxygen-poor blood from the fetus back to the placenta. We start off our journey in the placenta, which is just a highly vascularized organ that's found within the uterus of a pregnant woman. Within the placenta, we see a back and forth exchange of gases, nutrients, and waste products between the mother and the fetus. So essentially, this placenta is responsible for giving the fetus all of the gases and nutrients that it needs to grow, and at the same time, it's also responsible for taking all of the waste products away from the fetus and giving them back to the mother's blood where she can then expel those wastes. From the placenta, our oxygen-rich red blood cells are then going to enter the umbilical cord. So the umbilical cord is just a cord that's going to tether the fetus to the placenta itself, And making up this umbilical cord, we have two umbilical arteries and a single umbilical vein. So, are the oxygen-rich red blood cells going to travel through the umbilical arteries or the umbilical vein? Great, so you might have noticed in this image that the umbilical vein is colored in red, indicating it's more oxygen-rich than the umbilical arteries. The umbilical vein is going to take oxygen-rich blood from the placenta towards the fetus, and on the other hand of this, the umbilical arteries are going to take oxygen-poor blood from the fetus and back to the placenta. This is a great example that proves not every vessel colored red is going to be an artery. Remember, we can think of artery as standing for A away, or arteries take blood away from the heart, while veins take blood back towards the heart. The umbilical vein is going to take the oxygen-rich blood towards the neonatal liver, but not all of the blood is going to be delivered directly to the liver. The ductus venosus is a vessel that's going to act as a shunt and allow the oxygen-rich blood from the umbilical veins to bypass the liver and enter into the inferior vena cava. At this point, the oxygen-rich blood from the ductus venosus is going to begin mixing with the oxygen-poor blood of the inferior vena cava. Once it's mixed, we end up with some blood that's slightly oxygen-rich heading back towards the heart. So why bypass the liver? What's the purpose of having ductus venosus? And great, so there's a few reasons why we would bypass a neonatal liver. The first of which is that the liver is only semi-functional. In addition to this, recall that a fetus is getting its nutrients from a mother. In other words, a mother who has a functional liver who has already made safe the nutrients that she's going to send to the fetus. In addition to this, it would be kind of silly to send all of this oxygen-rich blood directly to the liver when there's organs like the developing brain that need it much more. After the slightly oxygen-rich blood has passed through the inferior vena cava and into the right atrium of the heart, most of this blood from the right atrium is going to pass through a small hole known as our foramen ovale. The foramen ovale is a small hole that links our right atrium with our left atrium. Foramen ovale allows for the slightly oxygen-rich blood of the right atrium to make its way into the left atrium, where it can then be ejected into the left ventricle and then ejected to go to the vessels that nourish and supply the brain. You might have noticed that in doing this, this blood has completely bypassed the pulmonary circuit. So what's the reason for this? Great, so remember that a fetus is in a fluid environment and thus has non-functional lungs as it relies on its mother for a source of oxygen. Because of this, it's much better to send this slightly oxygen-rich blood from the right atrium through foramen ovale into the left atrium, where it's then pumped down to the left ventricle, out the aorta, into vessels that will eventually nourish the developing brain, where that oxygen is really critically needed. This foramen ovale typically ends up sealing later on, but in some people it doesn't seal, and this is called having a patent foramen ovale. Oddly enough, this is more common than you think because in many cases, having a patent foramen ovale doesn't really cause any issues to the patient. So up until now, we've been talking about the pathway of oxygen-rich blood into the fetus from the placenta, but what about the pathway of oxygen-poor blood? What's going to happen to this blood that's coming from our superior vena cava? So this is really counterintuitive. You would expect that the blood from the superior vena cava, which is oxygen poor, and the slightly oxygen-rich blood from the inferior vena cava would end up mixing in the right atrium. Oddly enough, only a small amount of mixing takes place, and the majority of the blood from the superior vena cava doesn't pass through the foramen ovale. The majority of the blood from the superior vena cava is going to enter the right atrium, where it's going to then be ejected into the right ventricle. In other words, only a very small amount of blood from the superior vena cava is going to be allowed to pass through our foramen ovale into the left atrium. Now the problem with this is, isn't the blood in the right ventricle just going to go through the pulmonary circuit and make it back to the left atrium anyway? The answer to this is no. So in addition to the foramen ovale and the ductus venosus, there's one additional fetal shunt that we need to discuss, and that's our ductus arteriosus. This oxygen-poor blood in the right ventricle is going to be ejected into the pulmonary trunk here, and rather than head towards the pulmonary arteries in the lungs, this blood is going to pass through our shunt known as the ductus arteriosus, where it then enters into the descending aorta. We'll talk about why the majority of this oxygen-poor blood passes through the ductus arteriosus rather than going towards the lungs in another video, but for this video it's important to note the location of ductus arteriosus, and it's going to be sending in the oxygen-poor blood below or inferior to the vessels that would supply the brain. This is really ideal because all of the early branches off the aorta and the aortic arch are meant to supply the brain with that slightly oxygen-rich blood. On the other hand, we wouldn't want the oxygen-poor blood being sent towards the brain. Instead, ductus arteriosus sends the oxygen-poor blood into the descending aorta, where it's going to be mixed with that slightly oxygen-rich blood. At this point then, the descending aorta is filled with very slightly oxygen-rich blood, but this blood is still oxygen-rich enough to supply tissues other than the brain. Eventually this blood is going to enter into our umbilical arteries, and from here the umbilical arteries take this oxygen-poor blood back to the placenta where it can be reoxygenated, and we'll talk about why that occurs in a later video. Now we've talked about the major pathways of fetal circulation, so go ahead and act as if you're a red blood cell in the placenta that's been oxygenated, go to the fetus, and return back to the placenta. First, we start off our journey from the placenta. We then enter umbilical veins that will take us directly to the fetus. The umbilical vein then will lead us towards our neonatal liver, but remember, most of this blood is going to bypass the liver through a structure known as ductus venosus, which then drops us into our inferior vena cava. At this point, our slightly oxygen-rich blood is going to be sent up towards the heart where it eventually reaches the right atrium. From the right atrium then, it's going to pass through a small hole known as foramen ovale into our left atrium. From here, the slightly oxygen-rich blood is going to enter our left ventricle where it's then going to be ejected into the aorta. Once in the aorta, this blood can then go nurse the developing fetal brain. Now to discuss our pathway of the oxygen-poor blood, starting at our superior vena cava. So from the superior vena cava, remember that the majority of the blood is going to enter into our right atrium and then pass into the right ventricle. Once the right ventricle has ejected the oxygen-poor blood, it's going to enter into the pulmonary trunk. And rather than going towards the pulmonary arteries, the majority of the blood is going to pass through this duct known as ductus arteriosus. Ductus arteriosus dumps this oxygen-poor blood into the descending aorta, and after being in the arterial circuit for a while, this oxygen-poor blood will encounter umbilical arteries. The umbilical arteries will take this blood back towards the placenta. While the pathway of fetal circulation can be really tricky at first, if you draw it out like this, you'll find it's just like telling a story. Now that we've finished with our lesson, let's go ahead and do a mini quiz. Which of the following is in contact with oxygen-poor blood? So to start off this one, let's recall the pathway of oxygen-rich blood from the placenta. First, it was going to pass through the umbilical vein, so D is incorrect. From here, this blood headed towards the liver, but a large portion of this blood was going to bypass the liver using the ductus venosus, so B is incorrect. From the ductus venosus, this blood enters the inferior vena cava and eventually the right atrium. Once in the right atrium, this slightly oxygen-rich blood is going to attempt to move from the right atrium to the left atrium utilizing the foramen ovale, making C incorrect. So for the oxygen-poor side of fetal circulation, recall that the oxygen-poor blood enters from the superior vena cava and into the right atrium. This oxygen-poor blood then enters the right ventricle where it's pumped into the pulmonary trunk. Rather than sending this to the lungs, it's better to send it through our ductus arteriosus, where it can bypass the lungs and enter into the descending aorta. Now that we've had our mini quiz, let's go ahead and do a few review questions to see what you learned today. First, describe the flow of blood from the placenta to the fetus back to the placenta. Next, what is the purpose of ductus arteriosus? And lastly, what is the difference between umbilical arteries and umbilical veins? If you can answer these questions, you're well on your way to success. Everybody be sure to have a wonderful day, and I will see you in the next one. Hi everybody, Kelton here. Today we're going to be discussing the fetal circulatory system, so pause the video, take a look at the outline, and let's jump right in. In a previous video, we talked about the patterns of fetal blood flow and how one of these unique circuits was the act of bypassing the lungs. So, as a quick review, what shunts are used to bypass the fetal lungs? Nice, so the shunts that allow for fetal blood to bypass the lungs are foramen ovale, which if you remember is just a hole between the left and right atria, and our ductus arteriosus, which connects our pulmonary trunk to the descending aorta. As a quick review, recall that slightly oxygen-rich blood from the inferior vena cava is going to make its way into the right atrium, where it eventually passes through the foramen ovale into the left atrium, thus allowing this slightly oxygen-rich blood to bypass the pulmonary circuit. The ductus arteriosus, on the other hand, largely deals with the oxygen-poor blood from the superior vena cava going through the right atrium and into the right ventricle, and eventually to the pulmonary trunk, where it then passes through the ductus arteriosus into our descending aorta. So what are the physical principles that makes blood flow in these patterns? It's not as if red blood cells can decide where they want to go or have a mind of their own, so what mechanism is pulling them the way they're going? Great, so recall that a fetus is in a fluid environment in utero. In other words, the lungs aren't functional and they're not doing any gas exchange. Because of this, the lungs are really small and the arterioles and capillaries are very constricted and don't allow a ton of blood in them. This causes the vessels in the lungs to have a really high resistance, and blood is basically just like water. Water is always going to flow down the path of least resistance. Our blood is going to be the same. In the case of our oxygen-poor blood in the right ventricle, it's going to be pumped into the pulmonary trunk where it will then flow down the path of least resistance. There's a lower pressure in the descending aorta than there are in the pulmonary arteries. This means the blood is going to bypass the pulmonary arteries completely and pass through ductus arteriosus. Because of the high resistance in the lungs, the right ventricle is going to have to work really hard and is under quite a bit of pressure. Because of this pressure, the slightly oxygen-rich blood from the inferior vena cava enters the right atrium and moves down its path of least resistance from the foraminal valley into our left atrium. So as you might have guessed, the pulmonary arteries of a fetus have a much higher blood pressure than the pulmonary arteries of an adult. After birth, though, the lungs become functional and the arterioles and capillaries begin to expand, and this is going to end up decreasing the pressure of those pulmonary arteries. Another thing that's a little bit odd with fetal circulation is that blood's going to end up returning to the placenta through our umbilical arteries. Notice that our umbilical arteries branch off our common iliac arteries, so what's going to make it so that most of the blood is going to flow back towards the placenta where it can get new oxygen, rather than just flowing down into the foot. And great, so within the placenta, we see the opposite of what was occurring in the fetal lungs. The arterioles and capillaries within the placenta are under very low pressure and are very dilated, meaning that there's very little resistance in the placenta. This extremely low resistance makes it so that a large portion of the fetal blood ends up entering into the umbilical arteries and returning to the placenta rather than just continuing on down the leg. And this is really useful because no matter what position the placenta is in or the fetus, this low pressure is going to bring back that oxygen-poor blood towards the placenta. In talking about the placenta, it's important to note that while the fetus is going to get many nutrients and oxygen from the mother this way, not everything can simply cross over the placenta. One prime example of this is that red blood cells from a mother and red blood cells from a fetus are not allowed to mix. And this is really important because this allows for a fetus and a mother to end up having different blood types. In this image, notice that we have vessels from our fetus which can be seen in our chorionic villi being surrounded by a pool of maternal blood. Separating these two is the embryonic chorion, which has the main job of making sure that some things from the mother cannot be passed on to the fetus, and some things from the fetus, such as red blood cells, cannot be passed back to the mother. While red blood cells are not allowed to cross over the embryonic chorion, there are quite a few things that are allowed to cross, and this can be really helpful in some instances and really harmful in others. Now we're going to discuss a very specific case of what can happen if a fetus's blood and a mother's blood end up mixing, erythroblastosis fetalis. So if we break down this name, we see fetalis means involving a fetus, erythro typically indicates red blood cells are involved, and blastosis means formation or development. So just from this name, we can deduce that this pathology is going to have something to do with the red blood cells of a fetus during development. Before we get into the small details of this, let's recall that some people are a blood type known as Rh negative, while others are a blood type known as Rh positive. Individuals who are Rh positive have a cell surface protein known as rhesus factor, while Rh negative individuals lack rhesus factor on the surface of their red blood cells. The important part of this is if you're an Rh negative individual, you have the potential to make antibodies against the rhesus factor protein. Now that we've reviewed that, let's go ahead and see how erythroblastosis fetalis can actually develop. In order for this condition to develop, during the first pregnancy, the mother must have Rh negative red blood cells, and the fetus must have Rh positive red blood cells. This is where it's really important that the fetus's blood and the mother's blood are not allowed to cross and mix over the placenta. If this were to happen, the mother's immune system would encounter that rhesus factor protein from the fetus's red blood cells and make antibodies against the rhesus factor protein. But remember, because of the presence of the embryonic chorion, this is completely fine as the mother's red blood cells and the fetus's red blood cells are not allowed to mix. However, during childbirth, many of the vessels within the placenta will actually rupture, and when they tear or rupture, they end up exposing the mother's circulatory system to this Rh positive blood. From here, the mother's immune system begins to make very large amounts of antibodies that will attack rhesus factor. This is typically not going to cause any negative problems for the mother or her firstborn, but any children after this will have to deal with serious issues. So why is that? Recall that we had previously said that the embryonic chorion allowed for the transfer of certain things from the mother to the fetus, with one of these things being antibodies. While this is sometimes a good thing as a mother can bolster her child's immune system with her own antibodies, in the case of erythroblastosis fetalis, it's really bad because the antibodies against rhesus factor are allowed to cross the placenta and head towards the fetus. These anti-Rh antibodies, which are present from the birth of the first child, are allowed to cross over the placenta and enter into the fetal circulation, where they begin attacking the red blood cells of the fetus. This attack leaves the fetus severely depleted in red blood cells, meaning that it's anemic. In many cases, the fetus becomes so anemic that it doesn't have enough red blood cells anymore to do proper gas transport, and it ends up resulting in the death of the fetus. Luckily, however, there is a way to prevent this from happening, and it involves giving the mothers who are Rh negative a series of antibodies before their first child is born. So in short, if we were able to wind back time to the time of the first child being born and give the mother an injection of these antibodies, we could make it so the mother would never even recognize the Rh positive antigens on the surface of the fetus's red blood cells and prevent the buildup of the anti-Rh antibodies. Now that we've finished with our lesson, let's go ahead and do a mini quiz. A fetus would be at risk of erythroblastosis fetalis in which of the following situations? Assume that each second born was preceded by an Rh positive firstborn. Right off the bat with this one, there's a few things that we can use to easily get rid of a few answers. First, remember that erythroblastosis fetalis never actually affects the firstborn child, so this means we can get rid of A as well as C. Next, recall that this condition can only occur with a mother who is Rh negative, meaning she's capable of making antibodies against rhesus factor. In the case of answer B, an Rh positive mother would never make antibodies against rhesus factor because she has rhesus factor on her own cells. This leaves us with the correct answer, answer D. If the mother had not been given an injection to prevent the buildup of antibodies against rhesus factor, her immune system would have attacked the Rh positive second-born fetus. Now that we've finished with our mini quiz, let's go ahead and do a few review questions to see what you learned today. First, what mechanisms allow for blood to bypass fetal lungs? Second, what draws fetal blood back towards the placenta? And lastly, when is a fetus at risk for erythroblastosis fetalis? If you can answer these questions, you're well on your way to success. Everybody be sure to have a wonderful day, and I will see you in the next one. Hi everyone, Kelton here. Today we're going to be discussing the lymphatic system, so pause the video, take a look at the outline, and let's jump in. When discussing the lymphatic system, one of the first places people will look for the lymphatic system is around a capillary. In looking at this diagram, it's important to note that we have an arteriole on the left and a venule on the right, meaning that this vessel connecting the two is a capillary. Recall that on the arteriole side of a capillary, there's a very high amount of hydrostatic pressure that's pushing fluid from the inside of a vessel to the outside of the capillary. On the venule side of a capillary, we see that there's an osmotic pressure that's going to be pulling in most of the fluid towards the capillary. However, it's important to note that not all of our fluid is going to be reabsorbed into this capillary, and a good deal of it is going to be sitting here in between cells. This fluid is known as interstitial fluid. So what do you think is going to happen to this fluid that's just sitting around in between cells, and what problems do you think you could have if you had that fluid build up too much? Great, so simply letting this fluid sit and build up in between your cells would end up causing a lot of problems like running out of blood plasma or really severe swelling. Thankfully, the lymphatic system comes into play. The smallest of the lymphatic vessels are our lymphatic capillaries, and these capillaries are very leaky. Because they're so leaky, the osmotic pressure at the venule end of a capillary bed is plenty to push all this interstitial fluid into our lymphatic capillary. Once the interstitial fluid has entered our lymphatic capillary, it becomes what's known as lymph. Now, lymph is just a combination of interstitial fluid, fat, proteins, and even bacteria, and we'll talk about why it has some of those things inside of it a little bit later on. Another key thing to know is that the amount of lymph that you're going to have is determined by how much of that plasma is going to be brought back into the circulatory system at the venule end of a capillary bed. If the capillary only takes up a very small amount of fluid, then there's going to be a lot of lymph, as the lymphatic vessels are going to have to pick up the slack and put that into lymphatic capillaries. The lymphatic system, despite being one of the lesser known systems, is really important, and it has quite a few different functions. Lymph and lymphatic vessels are a subset of the circulatory system because, as we just saw, they end up taking the leftover interstitial fluid and returning it back to the systemic circulatory system. In addition to this, a few of these lymphatic vessels are also really important to the digestive system. And lastly, our lymph nodes, adenoids, spleen, and thymus are all parts of the lymphatic system that are really important to our immune system. Now we're going to talk about all of these in a little bit more detail later on. Other than just picking up interstitial fluid, our lymphatic capillaries have a very specialized role in our small intestine. Our small intestine contains small finger-like projections known as villi. These villi act to increase the surface area of the small intestine, which allows for more nutrients to be absorbed. If we look at a cross-section of a villus, we see they are in fact a finger-like projection, and we see that they have three main vessels. They have a vein, an artery, and a lacteal. As the green in this diagram indicates, lacteals are a type of lymphatic capillary that acts to absorb dietary fats in the small intestine. Lacteals then continue to drain into larger and larger lymphatic vessels until the fat has eventually made it back into the circulatory system. So as the lymph from lacteals or other lymphatic capillaries begins to flow into larger and larger lymphatic vessels, eventually we might encounter these things known as lymph nodes, which are these little bumps on this diagram here. These lymph nodes are filled with tons and tons of immune cells known as lymphocytes. These lymphocytes look over the incoming lymph, and if they encounter anything foreign like a bacterium, they end up triggering an immune response. In other words, they act as little police checkpoints to make sure that nothing harmful like a colony of bacteria is trying to take root in the very slow-moving lymph. If you've ever been to the doctor when you're sick and they've touched your neck like this, it's likely to feel for any swollen lymph nodes, because during an infection your lymph nodes will sometimes swell up and become a little tender and painful. In addition to this, adenoids, the thymus, and the spleen also have an important immune function. Lymph has a pretty long journey to take to get back into the circulatory system depending on where you're at in the body. The lymph is very slow moving, and it's going to be traveling through multiple different checkpoints like the lymph nodes. Eventually though, this lymph is going to make it back into the circulatory system, which is really important because it's going to act to replace all of the fluid that was lost in the capillaries. Lymphatic vessels are not going to deliver their lymph directly to the heart and instead are going to deliver the lymph into two circulatory veins. Oddly enough though, the two lymphatic vessels that end up delivering the lymph back towards the circulatory system did not share the same burden. The right lymphatic duct is going to end up containing lymph from the right upper half of the body. It's then going to drain this lymph into the right subclavian vein. The majority of the body's lymph, however, is going to end up in the thoracic duct, which is going to drain into the left subclavian vein. Moving on then to some final things about the lymphatic system, and I think these final things can be summed up pretty well by asking the question, is the lymphatic system more similar to veins or arteries? Great, so the lymphatic system is much more like the venous system than the arterial system. The lymphatic system doesn't utilize a central pump as it's not connected to the heart like the arteries are. Lymphatic vessels do have a small smooth muscle layer that helps to push the lymph back via peristalsis, but they're under very low pressure just like veins. Because of this, they utilize valves just like veins did to prevent backflow, and they also utilize the skeletal muscle pump to get the lymph really moving. To top it off, most of our lymphatic system is going to be mirroring our venous system, meaning that the lymphatic vessels typically like to follow where the veins go. Now that we've finished with the lesson, let's go ahead and do a mini quiz. Lymph from your right foot will pass through which of the following on its way back to the heart? For answer A, remember that any interstitial fluid that is not reabsorbed by a capillary is going to enter into the lymphatic vessels, not a venule, making A incorrect. Looking at answer B, the right subclavian vein is going to receive lymph from the right lymphatic duct. Remember, the right lymphatic duct only collects the lymph from the upper right part of your body, making both B and C incorrect. Answer D is our correct answer. Even though it may seem odd, the thoracic duct is going to collect all of the lymph from your right lower limb. Now that we've had our mini quiz, let's go ahead and do a few review questions to see what you learned today. First, what are the functions of the lymphatic system? Second, what are lacteals? And lastly, what are some things that may affect the amount of lymph? If you can answer these questions, you're well on your way to success. Everybody be sure to have a wonderful day, and I will see you in the next one.